Look at me, I'm the best looking glucose there is. Whoops! We have all the energy from glucose and the electron packets. I'm the energy guy who moves the H+ to the other side of the membrane. Let me in! Let me in! Let me in! Let me in! Let me in!
Cellular respiration is the chemical process that releases the energy stored in organic molecules of food in form of ATP. In cellular respiration, glucose is completely oxidized or broken down, releasing CO2 as the final product. Oxygen acts as the final electron acceptor and combines with hydrogen to make water. A total of 34 to 38 ATP are released. Cellular respiration can be broken down into three major steps: glycolysis, Krebs cycle, and oxidative phosphorylation. This is a diagram that shows an overview of the process. Glycolysis, which takes place in the cytosol, breaks down glucose to pyruvate. Pyruvate then goes into the mitochondrion, and a derivative of it enters the Krebs cycle. Both glycolysis and Krebs cycle produce ATP through substrate-level phosphorylation. The energy in glucose is transferred to electrons carried in NADH and FADH2. These NADH and FADH2 then goes into electron transport chain and produces ATP through oxidative phosphorylation. Let's have a detailed look at glycolysis. In the first step, a phosphate from ATP is transferred to glucose to make glucose 6-phosphate. Then glucose 6-phosphate is transformed into fructose 6-phosphate. Another ATP is used to add a phosphate group to carbon 1 to make fructose 1,6-phosphate, which then split into two 3-carbon molecules known as G3P. You will notice in the step so far, 2 ATP molecules have been used. An inorganic phosphate is then added to G3P, and in the process, NAD+ turns into NADH. Notice that this new molecule has phosphate on each end. A series of reactions take place in which the phosphate groups are transferred to ADP to make ATP. At the end of glycolysis, 2 net ATP are produced, as well as 2 NADH. The pyruvate then goes from the cytosol into the mitochondrion. Several enzymes catalyze the conversion of 3-carbon pyruvate to 2-carbon acetyl coenzyme A, yielding a molecule of NADH and 1 CO2. The acetyl coenzyme A enters the citric acid cycle. During the Krebs cycle, oxaloacetate joins with acetyl coenzyme A to form the 6-carbon citrate. Then the 6-carbon citrate will give off a carbon dioxide and generate NADH to form a 5-carbon alpha-ketoglutarate. The process repeats with another carbon dioxide given off and NADH produced to form a 4-carbon molecule succinyl coenzyme A. Then this goes through a number of steps of remodeling in which ATP, FADH2, and NADH are produced to form back oxaloacetate, and the whole Krebs cycle begins again. By the end of the Krebs cycle, all the energy of glucose is transferred to high-energy electrons carried by NADH and FADH2. This is an overall view of what will happen during the electron transport chain. The NADH and the FADH2 that carry the high-energy electrons will pass the electrons to the protein complex located in the mitochondria inner membrane. As the electrons move from one protein to the next, energy will be given off, and oxygen will be the final electron acceptor. This energy will be used to carry H+ from the matrix of the mitochondria across the membrane to the intermembrane space. This creates an H+ gradient across the membrane. During chemiosmosis, H+ diffuses back through the ATP synthase, and the process generates ATP.